to Boston. I'm Elise Gellerman, CEO of the Neuroendocrine Tumor Research Foundation, and it really is my pleasure to welcome everyone here this morning. This is our 2023 Margie and Robert E. Peterson Neuroendocrine Tumor Research Symposium. That's a mouthful, but we have a lot of good work that we're going to hear about over the next 2 days. This symposium is clearly our scientific highlight for the year. Our reach is truly international, more so I think than ever before. You represent more than 50 premier research institutions around the world and 12 countries. So for those of you who have traveled far and wide to be here, we are really grateful for your participation. We couldn't do what we do without you. We also have a number of friends of the foundation who are joining us here today, and we are grateful for all of you. NETRF has invested $36 million in research since 2005. As you know, NETRF funds innovative and proof-of-concept projects that other organizations do not fund. With a focus on basic and translational research, we have supported NET researchers to advance our understanding of neuroendocrine tumors and contribute to the development of new and improved treatments. And I know we're going to hear a lot more about that during this meeting. NETRF has also focused on recruiting scientists to this field by providing seed funding so they can investigate novel approaches to this challenging disease. And as I look back, because I think this is about my 7th symposium, I see a number of you who started as postdocs back then, and now you're running your own labs. That to me is a metric of success for the funding we've provided and the community that we have also provided for many of you. That's a really exciting thing for us. NETRF is very proud of our total impact on the field of NET research. I want to share a couple of results with you for just a moment. Since we began, we have funded research that has led to 20 clinical research projects, including clinical trials. Since 2017, when we took a look at some of our data and what many of you report to us in your progress reports, which is very important, keep up the reporting, NETRF-funded research has resulted in 8 patents, 5 industry partnerships to bring new technology or treatments to patients, 80 collaborations between researchers. Again, we saw some of that happening last night with the discussion with some of you. One of the metrics we're most proud of, 100% of the early career researchers that we have funded since 2017 are still in research, and 90% of them are still in NET research. So thank you for that persistence and that dedication to this field. So we know that there is much more to learn. That's why we're here this week. Patients are depending on our commitment to research, and they're excited to learn that all of you are working on their behalf. I'm always impressed by the discussions and the collegiality at this meeting. We appreciate the feedback we get from all of you that this is one of your favorite meetings of the year. That's very important to us. So this symposium is very special in the embodiment of not only NETRF's work, but your work. From all of us at NETRF, our board and our staff, on behalf of patients who are living with neuroendocrine cancer, thank you for your expertise and collaboration. I look forward to the sessions over the next 2 days. I want to give special thanks to the NETRF team. This symposium is a team effort. I think many of you have met all of our staff since last evening. This effort is led by Dr. Anna Green, our Director of Research, who you will hear from in just a moment. She joined our team this year and has brought us considerable expertise and commitment. So I'm happy to introduce her so she can provide some additional information to you about this meeting. Anna?
All right. Hi, everyone, and welcome. It's really great to meet you all. My name is Anna Green, and I'm the Director of Research here at NETRF. This is my first time planning and first time attending this symposium, and so I'm really happy to be here and to meet you all. But first, I really need to start with a thank you So I really must thank all of the NET researchers here today who took time out of their schedules and away from your family and friends and other commitments. And we really appreciate you being here today. I'd also like to thank you for your commitment to NET research so that we can learn more about this disease. I need to thank our board of scientific advisors. Thank you for being here. Thank you for helping us with our scientific program. And thank you for helping us moderate the sessions here today. I also want to thank our board of directors for holding us true to our mission to help patients and families and stay committed to funding impactful research. So thank you for being here today. And we have some special guests of the foundation here today, so thank you for being here. I hope it's a wonderful and productive meeting for you. And of course, the staff. I echo Elisa's sentiment that this is a very, very large team effort to put this symposium on, and I really thank all of the staff for their support every day, and in particular for this symposium. So if you need any help during the symposium, feel free to let me know, but you can also reach out to any of our lovely staff members featured here. And so with that, I'm going to turn it over to Chrissie Thirlwell and James Bibb to lead off the first session. Thank you.
Good morning, everyone, and welcome. It's so nice to see so many of you, faces that are familiar, and also all of you that are coming for the first time. I welcome you, and I thank NETRF and Elise for being able to run such an outstanding organization and provide this format for us to pursue our interests in doing something about these diseases. My name is James Bibb. I'm the chair of the Department of Translational Neurosciences at the University of Arizona College of Medicine in Phoenix, and my co-host will be Chrissie Thirlwell, and she's the dean of the University of Bristol College of Medicine across the ocean. Neuroendocrine cancers have presented unique clinical and translational research challenges for decades. However, in just the past few years, we've witnessed major technological advances in detection, drug discovery, and precision medicine like never before. Computational sciences, medical services, clinical trials, and basic research have been boosted with all the progress in high-throughput genetic and gene expression analysis, and artificial intelligence offers the promise of taking us to a whole new ballgame. The outstanding progress of exceptional research teams highlights this morning's presentations, which focus on personalized medicine. And with that, I'd like to introduce our first speaker, Emily Bergsland, who will talk about enhancing drug development for neuroendocrine tumors, a novel radiometric signature to predict survival in patients enrolled in the Alliance A021202 clinical study. Welcome.
Okay, thank you so much. Well, first off, I want to thank NETRF for the opportunity to speak to all of you today. And I would like to say that the work I'm going to present is really on behalf of a large study team for this clinical trial, and specifically Laurent Derkla, who has done the majority of this work. So why do we need different or advanced or improved radiologic endpoints for clinical trials? Well, the main one we've been using for years now is simply based on tumor measurements of size, and it's called RECIST 1.1. And one of the real challenges in our disease specifically is that most of our drugs that have been FDA approved are what are called cytostatic, meaning that mostly in well-differentiated tumors, they cause stability in tumor size rather than dramatic shrinkage. So it presents some challenges in really definitively defining what is efficacy if you're not seeing major shrinkage. And this is further compounded by the fact if you look at the definition of stable disease, it includes everybody from minus 29% tumor size up to plus 19% tumor size. And I think you can see from this sample waterfall plot, which is from a study from a few years ago with bevacizumab, which was a negative study because it didn't have improved activity compared to the control, which was interferon. But what you can see is there really seem to be two subsets of patients treated with bevacizumab. Some had growth straight through it, but they were still counted as stable disease. And then others had, that would be this group here in yellow. And then this group here, also in yellow below the curve, the zero line, they had actually a reduction in tumor size and probably did benefit potentially from that therapy, but it counts as stable disease. You don't see major shrinkage, and they're all encompassed in the same group. So I think stable disease is just a very heterogeneous group of patients, and it doesn't really tell us within that group who might actually be benefiting from a cytostatic agent. The other big problem in neuroendocrine tumors is that they can be very difficult to actually measure by CT scan. And a lot of these tumors are very vascular. CT is done taking pictures at various time points related to the contrast administration. This is the same patient on these images here with the pictures taken at various time points relative to contrast administration. And what you can see is that there are real differences in how well you can visualize the tumor between these different scans. And we deal with this all the time in clinic, where we have patients who get scans done on different machines, different centers, at different time points relative to contrast administration, and it can cause real challenges related to measuring the same tumor over time in a patient. And just to give you a real-world example of this, the RADIANT-2 study was a randomized phase 3 study of everolimus in functional extrapancreatic neuroendocrine tumors. And this was looking at everolimus versus placebo. And the primary endpoint was progression-free survival by central review. But the way this study was done, the central read was done later on. So after the study was done, they did the central read. And what they found is that the central readers didn't agree with the local readers, so the people who were doing the regular reads along the way. And in fact, when they came time to look at their data, in about 20% of cases, the central readers felt that the local readers had called progression too early. So patients had gone off study too early in retrospect, according to the central readers, and they couldn't use those patients' information for the primary endpoint. So that's called informative censoring, and they lost about 20% of their events, and ultimately they lost power and they weren't able to meet their statistical endpoint for significance, and it was a negative study. So this was an example where this problem of reliably imaging and agreement between radiologists was a significant one. And we were aware of this when we designed this study, the A021202 study, which was a randomized study of pazopanib versus placebo in extrapancreatic neuroendocrine tumors. And we made the central review real time, so that way when the local readers found progressive disease, it was sent to a central imaging office where they then had to confirm progression before the patient came off study. And the primary endpoint was progression-free survival by central review. Now, what we found was the drug did delay progression by about 3 months, and this was statistically significant. And interestingly, the local read found the same result. So there was concordance in the overall benefit of the drug in this case. However, what was really interesting when you actually drilled down on a patient level, there was quite a bit of discordance, but it was in both directions. And about over half of patients had discordance of their reads when you looked at individual scans and compared the central read with the local read. But in about 20% of the cases, it was a situation where the central read thought that the local read was about to take the patient off study too early. But in this case, because it was done in real time, they could give that feedback to the site and the patient could stay on study. So we didn't lose those patients in terms of their data. But in about 30% of the time, by the time the central read was given the possible progression, they agreed with progression, but they actually thought in retrospect it had actually happened earlier than the local team realized. And so those are patients that are at risk for potentially being overtreated with a study agent. So again, problems in both directions with potential undertreatment or overtreatment due to challenges related to imaging. So we had planned when this study was designed to bank these images and to look at them more carefully to start exploring new radiologic endpoints. So I'm going to be presenting very, very, very early data. It's very preliminary. It needs to be validated in larger sample size. We are looking at a number of endpoints, including things like tumor growth rate and tumor volume, but I'm just going to be talking a little bit about some of the preliminary findings regarding radiomics. So what are radiomic features? These are additional features of a tumor that are embedded within a standard of care image on a CT scan in this case, but are not visible to the naked eye. So you need advanced imaging software to pull out and extract this information. And there are many different types of radiomic features that you can extract, and you can use these features to then start looking at things non-invasively like the tumor grade, predicting for outcome, even predicting for response to therapy in some diseases. Now, this field is really in its infancy in the neuroendocrine space. On the right, you can see a number of publications that are starting to come out using radiomic features in neuroendocrine tumors. And the number of publications in the bottom right, you can see is going up very rapidly just in the last few years. So our collaborators at Columbia really were pioneers in the field of radiomics. Larry Schwartz is now at Sloan Kettering, but continues to collaborate with us on this project, along with his another colleague at Columbia, Laurent Derkla. So I want to walk you through the methodology very briefly. You start out, in this case, there are about 170 patients enrolled on the study, but the first thing you have to do is decide whose cases you can use for the radiomics. And because we're looking at changes in the imaging features over through the first scan, which was done at 3 months, you had to have patients who got to 3-month scans. So that's an important thing to say. So you're going to lose some patients who went off study early. Some patients, we had 10 patients who never started treatment. We had some patients that turned out not to have measurable disease, meaning they had a pleural effusion or some other sites that we couldn't easily measure. And so we lost almost 50 patients from our original 170 that we couldn't use for the analysis. That left 120 patients that we could use, and those were split between a training and a validation set for the purpose of this analysis. From there, what you can do is you can generate a 3D image, 3D rendering to look at things like tumor volume, and then you can extract these radiomic features using machine learning techniques. And that leads to something called we're calling a radiomic score, or RAD score. Now, from there, I'll talk briefly about you can start introducing clinical features as well, such as the primary tumor site, the arm of treatment, the sex of the patient, et cetera. And again, using an AI-driven approach, you can generate a signature that includes the radiomic features plus the clinical features, which we're calling an AI signature. And I should just say that this whole infrastructure is trained on data, about 3,000 patients worth of data that our collaborators have from cancers treated with a variety of different agents. That's used to train this model as we're trying to apply it to neuroendocrine tumor patients treated with this drug. So a couple of comments about this before I show you the data. The radiomics themselves, the signature didn't seem to vary by the other, looking at the other components of the AI sig. So it didn't vary significantly by primary site or arm or therapy or age, for example, as I show here. But we did look at the input or the impact of each of these different factors on overall survival in general as individual components. And one thing I want to point out is age and the primary tumor site were significant in terms of impacting survival. And this is known, this is not a surprise at all, but I want to point out that primary tumor site is highly significant. But what's interesting is when you look at the radiomic signature and the impact on survival, it actually is very dependent on the primary tumor site. We saw that patients with primary tumors originating in the small bowel definitely had a much better survival overall compared to patients who didn't have a primary tumor in the small bowel. But the signature itself was more informative for the non-small bowel tumors. So here you can see a survival curve, which in Black, you can see the small bowel tumors, and in those patients, it didn't matter whether they had a high or low radiomic signature. They had a good outcome regardless. But when you start looking at the non-small bowel tumors, that's where you start seeing the radiomic signature can differentiate the patients with expected better survival compared to the ones with the high radiomic score. So in this case, a low radiomic score was associated with a better survival, but just in the non-small bowel patients. So we also looked at the influence of age. This also impacts survival, and we found that when it's adjusted for age, it didn't change the outcome. The impact of the signature was definitely present in the non-small bowel tumor patients. Now, in terms of looking at PFS, this is very preliminary. It's small numbers for this type of study, so I want you to interpret this with caution. But we found very similar results for the non-small bowel patients, meaning that the radiomic signature was prognostic, meaning if you had a low signature, you had a better outcome, whether or not you were treated with pazopanib or placebo. It didn't matter. But the interesting finding was for the small bowel tumors, we found something different, and this is, again, very preliminary, but what it almost looks like is that the pazopanib-treated patients, you could identify patients likely to benefit from the pazopanib. That's the top red solid curve. Those are patients with the high radiomic signature that seem to do much better than patients with the low signature. So this type of finding suggests it could be predictive for benefit for pazopanib, but only in this organ site. So it's very complicated and still very early days, but very interesting nonetheless. And then you can take this data, the radiomic data, and you can integrate it with clinical data and generate this AI signature using machine learning. And so now you look at, this is again, overall survival in the population, integrating the radiomics with the clinical features. And again, you can identify high risk and low risk groups of patients. So I want to end just by saying that this is very early days, but the idea is to non-invasively, using scans that we all have available clinically, using images and information from those scans to start extracting information that can better predict, hopefully eventually, for response to therapy or overall survival, which will hopefully, this type of thing will aid in our drug development process as we move forward. But very importantly, these kind of data absolutely need to be validated in larger studies. And we're very fortunate that the cabinet study has just read out, which is an even larger study of a VEGF-TKI in patients treated with cabozantinib. And we're hoping at some point we may be able to validate some of these findings in that study. So I just want to end by thanking all of our, of course, patients, families, collaborators for this work, Laurent Derkla specifically for helping to spearhead this project, and then NETRF for funding this work. Thank you.
Thank you, Emily, for that remarkable talk. The use of this technology is really promising. We have time for a few questions. If you can, please go to the back and use the microphones, unless they're going to be circulated.
Hi, Emily. Nice, really beautiful work. So I'm curious, when you have different clinical sites using different instruments to obtain their radiographic data and their images, how does that influence trying to consolidate all of that data with your imaging analyses? Does that affect it?
Yeah, I can tell you that we did a couple things. I can't answer the question specifically, like in terms of the precise impact of the different machines, et cetera, but the idea is in this study, we did limit this analysis to people getting CT scans. We actually excluded patients who had had MRIs. You can do radiomic features on MRI as well, but for this, we limited it to CT. And then for this study, actually patients, each site had to submit a sample scan at the beginning of the study. Like there was a protocol in place for how to do the CT scan, and they had to have their scan kind of vetted, their scan technique, before enrolling. So now this is not done in the real world, but one of the projects that's coming out of this is going to be looking at discordance, for example, in patients. and we have one colleague working on trying to better define why that happens. Is it in the choice of tumor they're going to measure? Is it the way the scan was done? We're trying to sort that out to better characterize it. But ultimately, this is a large amount of data. You can do this on a large number of tumors, and the hope is that this AI-driven machinery is going to be kind of weed through all of the bumpiness of these scans to help us more in an automated fashion assess efficacy.
Wonderful, thanks.
One more question.
Hi, Martin Kaplan from London. Great talk as always. I suppose there's a couple of points. One is, you alluded to it, the variance between different centers, because the timing of the scans there's papers already out there on the heterogeneity within and kurtosis and all the rest of it within CT imaging, but actually it varies between sites as well, and trying to standardize that in the terms of the timing of the arterial phase and so on, and how one gets around that in that AI process. The other is, it would be quite neat to tie in with biological markers as well, and whether or not you tie in the NET tests or something like that to see if there's any impact with that. And the third, obviously, is the utilization of somatostatin receptor imaging with the gallium dotatate PET as well, and how all of that combines, particularly if you're going down this AI route. One would see then having a bigger program of tying in the nuclear medicine, the biomarkers, as well as the CT.
Yeah, thanks. I think that's a great, those are all really great ideas. I think fundamentally, when we went into this, the idea is how do you speed up drug development? And I think this sort of big picture view of what's going on, I think, is very important for patient selection. But also in this case, because this relies on the 3-month scan, the idea is, can you determine at an early time point if that therapy is going to be beneficial to a patient? So I think they're both valuable. It's patient selection, but also once you're on a study, can you get an early readout that will predict for later, clear efficacy down the road to speed up the drug development process? But you're absolutely right. Thank you.
Thanks, Emily. And just for the purposes of time, we just need to move on now. So it's a great pleasure to introduce Christopher Heaphy. He's from Boston University School of Medicine. He's very well known to NETRF symposiums, and he's going to be presenting some new data around the detection of ALT via chromogenic in situ hybridization. So over to you, Chris. Thank you.
Well, thank you so much. I really appreciate the opportunity to be able to present some of our data today. So as we all know, pancreatic neuroendocrine tumors are the second most common neoplasm in the pancreas, and the incidence and prevalence of these tumors is dramatically rising over the last few decades. And patients can present very similarly, but can have very big differences in outcomes, disease progression, and therapeutic response. So we really believe that there's novel biomarkers that are needed still. So it's been 12 years or so since we've first really dived into the deep sequencing of pancreatic neuroendocrine tumors. Lots of efforts, by many people in this room, but what we've identified as a field is that MEN1, DAX, ATRX are definitely recurrently mutated in pancreatic neuroendocrine tumors. We also have identified that ATRX and DAX mutations lead to this phenotype called alternative lengthening of telomeres, or ALT. And so when a cancer really needs to be able to survive and keep proliferating, and it has two ways to maintain its telomeres. One is telomerase, which I think everyone has heard of, and then the other one is this other pathway called ALT. At the time, we really thought that maybe loss of ATRX and DAX function would repair the heterochromatic state of the telomeres, and this would lead to aberrant homologous recombination specifically at the telomeres, thus facilitating ALT. Of course, it's much more complicated than that. There's been numerous studies by different groups really looking into this. And what we've identified and others have is that it's really driven by a break-induced replication pathway. And so you can see here that loss of ATRX or DAX leads to altered telomeres, and then either through replication stress, DNA damage, telomere erosion. This can lead to break-induced replication, specifically driven by RAD52 and Pol delta, and can lead to telomere extension. The one thing, though, is that this really leads to very kind of a messy telomere phenotype. So some telomeres are extremely long, some telomeres are extremely short, and we take advantage of that to be able to look at it in tissue. So we've developed previously, and we've been using this assay for multiple different years. is a telomere-specific FISH assay. So you can see here the telomeres are in red, the centromeres are in green. It's a control probe. And what you can see on the far left is an ATRX and DAX wild-type case that's ALT negative. Hopefully you can maybe appreciate the small punctate red dots in the normal pancreatic duct. And you can see the tumor right next door to it is ALT negative. You can hardly see any telomeric signal, and that's because the telomeres are extremely short. However, in contrast to the ATRX mutated case or a DAX mutated PANET as well, you see hallmarks of ALT. And so these are the presence of these large ultra bright telomeric FISH signals, dramatic telomere length heterogeneity, presence of extrachromosomal telomeric DNA, and also this helps drive the process of chromosomal instability. Us and others in the field have definitely been looking at ALT as a prognostic marker, and I think it's very well established now that ALT is a strong prognostic marker, particularly in primary pancreatic neuroendocrine tumors. ALT is associated with larger tumor size, higher grade, advanced stage metastatic disease. And of course, with the association with ATRX and DAX, it's closely linked to alterations at either protein loss of ATRX or DAX or the presence of mutations. I do want to point out, though, that if you look at different studies, anywhere from 5 to 15%, there's discordance cases between ALT and the status of ATRX and DAX, and that might be because you have a mutation that leads to altered function of the ATRX-DAX complex leading to ALT. But when you stain for it by IHC, you're actually still detecting normal protein in the nucleus. So there are discordances, but it ranges anywhere from 5 to 15%. So because of all these different studies, we really wanted to put together a very large cohort study and really do this as broad as we possibly could. So we put together and have collaborated with different groups of international assessment of PANET biomarkers, at least tissue-based biomarkers. In this particular study last year, we looked at over 1,300 neuroendocrine tumors. 561 of them were pancreatic neuroendocrine tumors. It was a wonderful collaboration between groups in the US, Netherlands, Italy, and South Korea. And we evaluated ARX, PDX1, ATRX, DAX, all by IHC, and ALT by our telomere-specific FISH assay. Here it's shown in green, so you can have it labeled either in red or green, depending on which assay you're using. What we found, one of the major take-home messages of that particular paper is that ALT was the strongest biomarker for prediction of recurrence-free survival. So if you look in all pancreatic neuroendocrine tumors, again, a cohort of over 560 cases, you can see the ALT-positive cases have a shorter recurrence-free survival compared to the ALT-negative cases. And in a multivariate analysis, this is independent of all other parameters. We also did a subset analysis and looked at small pancreatic neuroendocrine tumors that are less than 2 centimeters, and we looked and evaluated close to 200 cases. Again, in this particular subset, only about 9% of the cases are ALT positive, but you can see the ones that are ALT positive definitely have a reduced free survival compared to the ALT negative cases. So at the time, us and others have come out with different reviews or perspectives in terms of trying to get this assay into a clinical setting and wondering if we can actually clinically implement it and was this assay ready for prime time. And what we came to the conclusion is that our telomere-specific FISH assay does have some limitations that can be overcome in a research setting, but maybe not in a clinical setting. So there is significant telomere heterogeneity, as you can see here on some of the images. It definitely requires a lot of technical expertise to be able to do the assay and then actually score the cases themselves. And there's sufficient cost. So you're talking about fluorescent probes, use of a fluorescent microscope in a clinical setting, and then you would need specialized staff to be able to make these calls. At the end of the day, we concluded that it was challenging to do that in a clinical setting, so we wanted to develop another assay that might be able to be used. So we collaborated with Atur Singhi at the University of Pittsburgh Medical Center, who's a pathologist, and we took our telomere-specific FISH assay, which produces really nice images that I like to look at, and turned them brown. So we turned it into a chromogenic assay in situ hybridization. And as you can see here, if you look at an ALT-negative or ALT-positive case, what you can see is there's obviously a big difference between the two. On the left in the ALT-negative case, you can even see kind of the background telomeres, small punctate dots, brown dots, and hopefully you can appreciate the difference between an ALT-positive case that you see on the right. We took that assay and then we compared it head-to-head with our FISH assay in a cohort of about 300 pancreatic neuroendocrine tumors. And what we found is there was 100% concordance between the new chromogenic assay and the FISH assay. As expected, using this ALT-CISH assay, we also see that ALT-positive pancreatic neuroendocrine tumors have reduced free survival. I think the big thing for this particular assay is it could be done in a clinical lab using commercially available reagents and also be used on FDA-approved autostainers. When you get a slide out at the end, since it's in a chromogenic, not in a fluorescent probes, a pathologist can actually score these and make the calls, and you don't need specialized staff to be able to do this. I think this really facilitates the clinical implementation of this particular assay. So I study telomeres in general in the ALT phenotype. So there's obviously pancreatic neuroendocrine tumors are enriched for ALT, but there's other tumor types that are also enriched. So this was a review we published a couple of weeks ago in which we looked at other tumor types. So a lot of the sarcomas have high prevalence of ALT. A lot of the CNS tumors do as well. So we wanted to be able to take our assay that we did in validated pancreatic neuroendocrine tumors and see if we could apply it to other tumor types. So we evaluated a cohort of surgically resected leiomyosarcomas that are enriched for ALT and found that, again, in this setting, in this tumor type, there's 100% concordance between the CISH, the chromogenic assay, and the FISH, and that we could use this. We also evaluated the prognostic significance of ALT in this, this tumor type, and we found that, again, ALT was a prognostic marker in leiomyosarcomas. Maybe not as strong as what we see in pancreatic neuroendocrine tumors, but this validates what other groups have seen before as well. So we've been able to develop a rapid and cost-effective chromogenic ALT assay that can be done in a clinical lab using FDA-approved autostainers. In this case, it was the Lycobond 3, because that's FDA-approved, and it can be done in a CLIA CAP accreditation pathology lab. We do think there's multiple advantages for this particular assay to be able to utilize this. I've shown you some of the implications in terms of prognosis, but we also think there's implications in terms of therapeutic ramifications as well. So if the ALT pathway is really driven by this break-induced replication repair, you can imagine that you can develop inhibitors that start to target this pathway. So it might be Rad52 inhibitors, Rad51, FANCM inhibitors. Another approach that we are also undergoing right now, and other groups are as well, is a synthetic lethality approach. So if you lose ATRX or DAX and you lose the function of that, those functions of that complex, it could also make those cancer cells more sensitive to potentially ATR inhibitors, WEE1 inhibitors as well. So we've been fortunate enough in the last year or so to be able to get additional funding to support this type of research. And right now we're doing spatial transcriptomic profiling on pancreatic neuroendocrine tumors, really comparing ALT-positive cases to ALT-negative cases to see what else is different, not only within the cancer, but the tumor microenvironment. And we're also looking at these genetic vulnerabilities in ALT-positive cases. I've been really lucky to collaborate with a really good group at BU and Boston Medical Center. So Ruben Dries is a computational biologist that's helping on all these studies. Everyone knows Matt Kulke. He's the section chief. Hussein Assi is a medical oncologist. Rachel Flynn is a basic telomere biologist. And so I think we're putting together a really nice group that we can actually start to tackle all these problems. And with that, I would just like to acknowledge all the people that worked on these studies. One of the things I really love in this field is the collaborations and how willing people are able to collaborate with each other. So I really want to highlight that, and then also thank the NETRF for funding this type of research for us. Thank you very much, and I'd be happy to take any questions.
Thanks very much, Chris. I can see we've got time for just one or two questions, but we will have time at the end for an open panel discussion.
Hey, Chris, that was great. Question, I'm intrigued by the heterogeneity in the telomere length or whatever between. Do you see the same thing if you knock this out in a cell line? If you knock out either of these components in a cell line, do you see the effects on the telomeres, and do you see that heterogeneity?
We do in certain contexts, so it really depends. So a lot of times we take a cancer cell line that's already using telomerase for its telomere maintenance, and then we knock out ATRX or DAX. In some situations, we definitely see that phenotype. In others, we don't. But we've already taken a cell line that's kind of like a normal cell needs to go down and then either pick telomerase or ALT. But if we also take a particular cell line and then knock out telomerase itself and either ATRX or DAX, we definitely see that strong phenotype. Yeah.
And you see this heterogeneity too in it?
You see the heterogeneity, yeah. And it's because it's a process that's ongoing in these cells, and these are byproducts. These large telomeric foci are byproducts. Some of them are actually attached to the end of the chromosome. Some of them are extracellular or extrachromosomal, I mean. Yeah. And so that's what we're picking up.
Thank you.
Sure.
Hey, Chris, really nice work. So I was curious about heterogeneity of the tumors, and you're qualifying them as either positive or negative. Do you have tumors where 90% is negative and 10% is positive? And how do you score those? And what do you think that means if you were to break down your positive samples and say, it was really only 30% positive? Do they do better than tumors that are 80% positive?
Yeah, no, it's a really good question. So one of the issues has been in the field is that it is positive or negative, and that our threshold for the detection of these foci in a tumor is actually 1%. So it's the difference between 0 and 1% and greater. So we see tumors anywhere from 1% to 80 or 90% of the cells, but it is that heterogeneity that we're picking up. We've tried to break down the positive into try to quantify, if it's lower or higher in terms of the heterogeneity. And we haven't seen any major differences with outcome there, but we might not have completely captured that. The only other heterogeneity you would see is if you have, every once in a while we do see subclonal, where you might have a tumor and then eventually have lost ATRX or DAX. But you'll definitely see that it's negative, positive in one area and then negative in another.
Outstanding.
Great, thanks.
Next, we have Nicholas Skill from Louisiana State Health Science Center in New Orleans. Hope I'm pronouncing that right. Neuroendocrine tumors, he's going to talk about neuroendocrine tumors. Ex vivo angiogenesis is a correlation for outcomes and survival in responsiveness to therapeutics.
Thank you, and thank you for the opportunity to present the data. So just to put this into context, I'm new to the neuroendocrine community. My background was in liver and hepatocellular carcinoma. My wife and I were recruited to LSU to take over from the program that was developed by Gene Waltering. So a lot of this data I'm going to show is from Gene's lab back in the day. So when I showed up, I found that he had an immense, huge biorepository of tissues. He had a very big database of his patients, and he was very, 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 very interested in angiogenesis. And he had developed an ex vivo assay where he would measure angiogenesis growth from tumors. So my job was to try and expand on this narrow focus and to utilize the resources that he had and to build collaborations and to grow the lab. So like every institution, we have weekly invited speakers. And I went to one, and there was a lady presenting her experience on the NCI Exceptional Responders Program, where you could send them tumors that had responded to chemotherapy, and they would provide you with omic analysis. And I was like, well, that's a great idea, because we've got all these tumors. Gene's been doing all this work looking at angiogenesis, doing pharmaceutical and cytomedicine challenges. So why don't we look at that and see whether or not we can develop some exceptional responders? At the same time, we had a med student come up to me. He said he wants to do some research experience, but he doesn't want to do any bench work. So, okay, fine. So we have this data. He has the time, so let's go do that. So, okay, so really three different aims. A, does the angiogenesis assay tell us anything? Does it correlate with overall survival? Is it just a random number that he's been generating for the last 20 years? The second one was whether or not it actually correlates with some intervention. So he did a panel of 20 different things, and we went through, and we decided we were going to look at SUTENT. And then the last one was, from that data, can we identify which of the tumors are actually exceptional responders ex vivo? So I don't actually even know whether any of these patients got SUTENT. All I know is that their tumors were analyzed. Okay? So I'm not going to go into Gene's assay much. If you want to read it, he patented it in 2002, and then he published a paper on it in 2010. There isn't a great deal of information out there, but most of the methodology is in these two papers. So just to be sure, this is really what I took from the claims in his patent. So basically, he was just saying, we're going to get a matrix, we're going to embed the tissue in the matrix, we're going to apply a medium, and we're going to let them grow for the next 2 weeks. And then after that, we're going to measure what growth there was. Okay, so this is an example. So each tumor had an N of 32. So 196-well plate could do 3 tumors. And so he measured 2 different things. He measured initiation, which was, did any part of the tumor have a angiogenic sprout coming out of it? And then the second thing was growth. And so he separated the tumor into 4 quadrants, and each quadrant had a scale of 1 to 4. And so if you're looking at here, A is a score of 1, B is a score of 2, C is a score of 3, and D is a score of 4. So each tumor fragment would have a maximum score of 16. Okay, and so the next thing he did some electron microscopy work just to show that there was a lumen inside of these and they weren't just vascular smooth muscle cell expanding. So as you see from the proximal to the tumor, way to the distal, you can see that it's changing. Okay, so there was a huge amount of variation. So in his original patent, he showed data for 58 tumors. So this is extended data for 338 tumors. And you can see on the left, we're looking at initiation. And so whether or not any sprouting occurred in the tissue. So it ranges from 0 to 100%. And so on the right, you have the same tumors in the same order, and they're looking at growth. And you can see that there's still a great deal of variation. So whether this is tumor variation, whether there's just interassay variation, whether there's operation variation, but it's not a particularly accurate assay. So then we look, well, what does that mean? So we picked 1 year, 2014, and we picked that because that was the year that our health system moved to Epic, and that was the system that was still available, so we could look at patient records. So we went back and we took each tumor that was analyzed in 2014, and there was 58 of them. And then we looked at whether or not the patient was still alive or whether they died. And then we did analysis whether or not there's a difference between patients that had a high angiogenic initiation or growth and ones that had a low. And we noticed that there was a statistical difference, but they're pretty much, very similar. So then we did some Kaplan-Meier curves, and as you can see, for initiation, tumors ex-vivo angiogenesis that had a high had a worse outcome for the patient, and ones that were low had a better outpatient. Okay. And again, we did the same for growth. And again, you see that the patients who had low growth did better than the patients that had high growth. And again, this is just ex-vivo data. But we're tracking back, we're trying to correlate back between the patient's outcomes and what we're seeing in the dish. Okay, so then we looked at SUTENT, and then everybody probably knows SUTENT. It's a very common thing, chemotherapy. And so this was one of a bunch of chemotherapies that Gene was using, and there we go. And the idea was this, is we're trying to pick out which one of the tumors had responded better than the others. And as you can see from this graph, we're plotting growth versus initiation, and we're trying to find ones where SUTENT is inhibiting both growth and inhibition. And so out of this, 6% of the tumors, you see at the bottom left-hand corner, you see the ones in red, and those are the ones that sort of match up with the NCI's classification of an exceptional responder. And again, this is ex-vivo. This is not in patients. I don't even know whether or not these patients ever got SUTENT. So we're a surgical center, so people come and see us and they have their surgery, then they go back to their home medical oncologist. And so their chemotherapy is not even in my chart, so I don't know. So whether I know that whether these 4 patients that were identified as being exceptional responders. Whether they even got SUTENT, I don't know. So what we want to do, though, is now take those tumors, because we have them in there in our biorepository, and we've sent them to the genomics and proteomics people at LSU, and they're going to try and look and see whether or not there is a profile that identifies them when you compare to tumors that don't. So that's it, really. So that's, at the end of the day, we're just trying to look and see whether they can stratify patients based on the tumors that we take out of them, and whether or not we can personalize their treatment plans and see whether or not they're appropriate for any particular angiogenesis treatment, and then try and develop biomarkers, because this ex-vivo assay is not particularly very accurate, and also it takes 4 weeks to actually get an answer. And so that was it. Okay, I'm done.
Thank you, and it's nice to see that Eugene's work is yielding results. I'm sure a lot of you are familiar with him as a longstanding member of this community. Are there any questions? I would just say that we're all looking forward to hearing how the genomics and single-cell analysis imparts better understanding of the determinants of angiogenesis and its relationship to responses to VEGF inhibitors.
That'll be next year.
Yeah, again,
Thank you.
Got to come back and tell us.
Thank you. It's now a great pleasure to introduce Iakovos Michael from Sunnybrook Research Institute, University of Toronto. He's going to be talking to us about establishment of novel patient-derived models for neuroendocrine neoplasms. So over to you.
Thanks so much. Okay. So I don't need my slides. So hi, everyone. It's my pleasure to be here. Thank you so much for the invitation. Okay, here is my talk. So I changed a little bit. We also have some additional data about organoids. So today, I don't think I have to introduce why we need new models in the field of neuroendocrine tumors. Really, thanks to the NETRF pilot award that we got last year, this allowed us now to establish a method to collect samples from patients that they come for surgery in our institute. Then these samples usually go to pathology, and then from there we split them into 3 for FFP for diagnosis. We snap froze some of these samples for biobanking, and then the rest of it will be diverted in my group as a first sample. And usually this happens every Wednesday afternoon. So then from there we take these tumors, and usually we split them into 2. Half of the tumor, we try to derive the patient-derived xenograft ex ovo, and I will explain what that is. And the other half, we use it to derive organoids, what we call PDOs, or patient-derived organoids. So you will see for the patient-derived ex ovo, we take tumor fragments 2 to 3 millimeters, and then we graft these ones in the choroidal membrane of dark eggs, and these are the tumors that we get at the end. And in the case of organoids, we will disassociate these tumors, we put them in 384 well plates, and then we will get the organoids, as I will show. So why are we using this method? So this method is being used a lot in the early days to understand angiogenesis. And what you see here in the bottom, sorry, I can point only to one screen. What we do, we receive the eggs at day 0. We incubate these eggs for 4 days, and then we crack the eggs, and we put here where you see the choroidal membrane, and in the middle the embryo in these petri dishes. And then we graft the tumor in the choroidal membrane. And we do this because, as you will see, this allows us now to do different drug treatments and also use ultrasound and visualize the tumor growth. So engraftment takes place at day 8, and then usually at day 12 we can start treatment, and we have to end point for this assay is at day 18 to 19. So that gives us around a week to do the treatment and at least understand the initial response of the tumor. So as you see on the top now, like in what's shown as panel B, this is the choroidal membrane, and this is a very highly vascularized structure, so that allows now for the tumors to get nutrients. So when we started, we wanted to see whether actually we can get any of the net cell lines growing in this assay. We started with some of the mouse cell lines we have in the group, and then we move into human pancreatic neuroendocrine cell lines. And what you see on the top, it's bright field images, and what is circled in red are these now cells growing and forming tumors in these coral endoids. And below now you see how we can image these tumors using ultrasound, and we can follow up the tumor progression as it goes. And so then we move into tumor fragments. First, we use one of the models we have in the lab. This is the RIPTAC model for pancreatic neuroendocrine tumors. We take tissue fragments, and again, what you see with H&E on the bottom right-hand side, I guess, is the tumor mass, and what you see on the outside is the CAM. Now, the real question was whether we can actually engraft any patient samples. So we started this, and one example here is a low-grade small intestine neuroendocrine tumor on the left. On your left side, you can see it's a primary tumor, and then from the same patient, now we have liver metastasis. On the top, again, you can see the bright field images of the tumors growing. In the middle, you can see now with the ultrasound, and with yellow, this is the tumor or the tumor graftment for primary and liver metastasis. And now what you can see below, again, using ultrasound now, we can actually see the blood flow in this tumor. And again, since we're using this ultrasound technique, we can both measure the volume, but also the vascularity in these tumors. So here now is the question is whether these tumors actually maintain the histology that you usually see from patients, and these were validated by the pathologists in our hospital. So just to orient you here, what you see around, it's the coral endoic membrane is highlighted with blue, and the rest of it, of the sample in the middle, this is now the tumor that we got from the patient. And this, again, a well-differentiated primary neuroendocrine tumor. So if we zoom in one of these areas, on the outside, you can see the avian CAM, this is the coral endoic membrane. What you see here is the avian blood cells. So in the dark, the blood cells actually are nucleated, so it's very easily to spot. And then below, you can see these tumor nets, which is the neuroendocrine tumor. What is really very interesting and I think very, very exciting is that the stroma is also maintained in these graftments. So this again from the same patient, but now this is a liver metastasis. And again, if we zoom in now in the middle of this liver metastasis, again, we can see these different tumor nets. But again, I think what's really exciting here now is we can see these red blood cells from the cheek embryo actually in the middle of this tumor. So what we think will happen is that we have anastomosis of the blood vessels from the initial specimen with the blood vessels in the CAM, and that allows now to get back the blood circulation in the specimen. This, again, some more histology. This is staining from chromogenic, which stains for the cancer cells. And again, in the middle now, you can see this stroma from the original tumor. So we've done this now. I think these numbers are from September with, I think we're about to 7 different patients. We use both primary lymph node metastasis and liver metastasis. We established, I think, now around 150 PDX ovos so far. And what we call the take rate, it's around at least 80%. The lowest one is from lymph node nets, but it goes up to 90% for the primary tumors. Now, okay, so what can we do with this other than looking at the histology? What we think we can use approach for is to now understand whether these patients will respond to different treatments. This is a different program that we have is for renal cell carcinoma. Again, we do the same thing, and we start treating these ones now. And one of the things I will point here is the nivolumab and atezolizumab. So these are immune checkpoint inhibitors. So what you can do now, you can go and you can inject this IV, and they go into the tumor, and then you can monitor the response. And now you see here now for this, again, in this water plot flow, each one of them is a different CAM, and you can see the response for nivolumab was better than atezolizumab. The other thing that, at least for renal cell carcinoma, was very interesting, you see in panel F, now we can also quantify the number of T cells, and these are the human T cells that they were residing inside the tumor. So now you can see in the vehicle how many T cells you have, but when now if we use the immune checkpoint inhibitors, you have expansion of the T cells that they were already came with the tumor graftment. So we hope that in the future we'll be able to do something like this with NETs. So I'm going to switch to PDOs, and that's also it's something that's very exciting for me. Again, from patients, now we can isolate cells, put them in 384 well plates, and then we use robotics for drug treatments, but also for imaging. These are some of the initial studies. We started with 500 cells, 1,000 and 2,000 cells. Now with plating 1,000 cells, we have almost 100 success of doing organoids, and we can do up to 1,000 organoids for each specimen we get. And these are some of the images. The other thing that is very exciting, what we can do now in our institute is we can use what we call these chromolives and different dyes that we can put in the organoids at day 1, and now we can both monitor how the organoids are growing, but we can also monitor the metabolism, the mitochondria function, the ER stress, and also apoptosis. So we can use these parameters to understand what the different drugs are doing and how the organoids are responding. So if you now go use this confocal microscope we use, and you can do cross sections, and you can start from the bottom of this one organoid. It is from the top, from the left to the right. You can see that actually these organoids, they form villi. So you have if you go in the middle section, you have the cells on the outside, and in the middle is empty. And you can see this even more clear here when we use the dye for the mitochondrial activity. So we started some experiments, and I think what was very reproducible in all patients. One of the advantages is that we always get much primary and metastasis samples. Always the organoids from metastasis, they are growing bigger, and I think that's something you will probably expect. But also this one example treatment with sunitinib. Now this from the same patient again, and you see how the primary tumor is responding to sunitinib. But when you look at the metastasis again from the same patient, now it doesn't respond to sunitinib. So we hope that we'll use this platform now to identify drugs that we can target metastatic nets and then go back to the PDX OVO to validate our data. So I think the conclusion is that I think now we have the platform in our institute for both establishing patient-derived organoids and patient-derived ex-ovos from low-grade well-differentiated nets. We can do that from both primary lymph node and liver metastases. And these methods obviously have different pros and cons. We want to characterize the microenvironment in these PDX ovos to understand more, and we're going to move on with some preclinical studies. The first one that we identified in our institute as priorities to find sensitizers for PRT treatment. So we are a very new lab. We're not less than 2 years, but these two amazing students, Dr. Nilashki Kulatunga is a postdoc, and Zoe is a PhD student in my group. They did all of this work in collaboration with a group of Dr. Hong Leong and Dr. David Andrews in my institute. I really have to highlight that without the clinical team, we wouldn't have been able to do any of this. And of course, the pilot grant I got from NETRF, it was the first grant I got in my group. And I wouldn't have been here today. Probably I would have been struggling writing grants. But really allowed us to kick off our group. So thanks so much. I'll be happy to take any questions.
Thank you very much. I thought it was an absolutely fascinating talk and really beautifully presented. I can already see people queuing up for questions. We do have a little bit of time, don't we? I think we can at least take the 3. So maybe Emily start.
Okay, yeah, really interesting talk. I was curious if you've had time to look at the uptake in your PDO model relative to organ primary site or proliferation rate. Is it simply that the highly proliferative tumors are easy to grow, or is there something more complex than that?
So in terms of proliferation, some of the primary tumors we had, it was 1%, and we don't have problem to have organoids. Now these organoids, it takes 4 weeks to grow, But for me, as a scientist, I like this because now it allows me to go at week 3 and start drug treatments or do any manipulations and I have time to evaluate that without having to passage the organoids. Mainly, we look for GI nets now. So all of our specimens were from small intestine or colon nets. We haven't had any pancreatic nets yet. Thank you.
Thank you for this fantastic talk, in my opinion. Am I correct that you mentioned that you had a very high success rate in generating PDOs?
Yes.
Because to my knowledge so far, I think the results have been quite discouraging in the scientific community. So are you using different methodology compared to what has been done in the past 5 years without any success?
I don't know if we used really any difference. So the 384 well plates we use, they have a gel cap inside, so it has the shape of a U shape, and the cap it's a extracellular matrix. So when we put the cells, basically you kind of force them to come together and they start aggregating. The media we use are always defined media, which we are still trying to improve. So no serum, definitely. But the success rate, I think we only had one sample that we didn't get any organoids, and we suspect that was mostly normal tissue. But when we have cancer cells there, so far it's being 100%.
But you mentioned the aggregation, so does it mean that it's not really the classical organoid model, but more a spheroid model or something like that, bringing together cells rather than outgrowth of stem cell-like cells? Because that could explain the higher success rate.
Yeah, there could be an explanation. I mean, the thing is, we always keep some cells behind, and we put them in a regular flask, and we always get organoids from there as well, so that we get these organoids forming. And I didn't show, but now we have around 10 lines that we call them long-term PDOs that we are growing. When we know more, I will be happy to talk about it and share anything we get from this.
Okay. Thank you.
Thank you.
Great talk, Iacobos. So I have a question, and maybe you are thinking about this or you address this. You're able to get tumor growth of the primary patient tumors in your chicken embryo system. Can you use that as a stepping stone to then take those fragments that have expanded in your system and then put them into mice as more of a PDX model that we can then study in that system?
I'm thinking we want to do it. Maybe that's kind of a first stage to get the tumors to adjust outside of the human body. Right. We are thinking of it. I think something I want to highlight that within this 1 week, what we're trying to do is to maintain the tumor graftment alive, and then hopefully see how the patient will respond to treatment. Again, if you have a primary tumor with 1% Ki67, actually does maintain that 1% Ki67, which is a good thing. So we're not really, which it would be wrong if we were now seeing Ki67 moving to 50%. I think we're just changing the biology. So we're not changing the biology. These graftments are still staying as if it's a 1% Ki67, will stay 1% Ki67.
Yeah,
but we're going to try that.
Are you increasing the angiogenic nature of the tumor, though?
That's something we're going to have to look into it. We're looking for markers to differentiate the endothelial cells from the duck embryo and the human. We are trying to solve this.
Great, thank you.
You're welcome.
Actually, I was going to take Chair's prerogative. We've got a couple of minutes left, so I was going to ask a question. That's okay.
Thanks
and it's fantastic to see the. Oh no, come back. I'm going to ask one. Come back. I'm not done.
Okay,
no,
sorry.
The end of that. I was just going to ask a quick question. It's fantastic to see the increased sort of take rate you described, so 80 to 90%, which I believe is around 40% in some of the other organoid systems we've seen. Have you got a sense of what we would call the doubling time of the tumour within your system, just to see how confident we are that it's reflecting the tumour biology? I think, you touched on that just now with Dawn, but it's just because there's quite a limited window, isn't there, for intervention in terms of the gestation in the egg system. So do you have a sense of that yet?
So the question was about the PDOs, the doubling time.
Yeah.
Yeah, and it's something we can definitely calculate because we image these PDOs once a week, so we can definitely figure this out. We haven't done it yet. We know that always the primary tumors, they grow slower, and we've seen sometimes with the patients, we know the primary is 1% Ki67, and the metastasis is 10%, and we can see that in the organoids as well. So at the end, the organoids from metastasis are larger. Going back to PDX over, I think that we're not changing that, so we stay very low, 5%.
Thank you. I'm sure we'll continue this discussion later.
Thank you so much. Thank you.
Okay, way cool. All right, our next talk is by Alexandra Sexton Oates, who's in the International Agency for Research on Cancer in Lyon, France, and she's going to speak about understanding the relationship between lung carcinoid histopathology and their molecular characterizations.
Thank you very much. So my name's Alex. I'm a postdoc in the Rare Cancers Genomics team at the International Agency for Research on Cancer, and we're based in Lyon, France. And I'm going to give this talk today on our NETRF-funded project, Reconciling Lung Carcinoid Histopathological and Molecular Classifications, and the PI for this project is Mathieu Faux. So I'll just give a teeny bit of background about lung NENs, which I'm sure everyone knows, but it's just to reiterate the importance of the histopathological classification. So lung neuroendocrine neoplasms, or LNENs, are rare diseases encompassing both low-grade, well-differentiated tumours called LNETs and the high-grade polydifferentiated carcinomas, or LNECs. So the current classification system by the WHO separates NETs into low-grade typical carcinoids and an intermediate-grade atypical carcinoid. And this is based on two key criteria, mitotic count and the presence or absence of necrosis. So the overall survival rates for lung NETs, are quite good, especially when you compare them with the carcinomas, but metastatic disease and post-treatment relapse do occur. The distinction between typical and atypical carcinoids is considered to be a critical prognostic factor because typical carcinoids generally have better outcomes than atypical carcinoids. So there's a median 10-year overall survival rate for typical of 89% and 51% for atypical. So in addition to this morphological LNET classification, we and others have shown the existence of molecular groups within lung NETs, and these are named carcinoids A1, A2, and B, as well as a fourth smaller group, just kind of a special group called supracarcinoids. And these are carcinoid tumours that have carcinoid-like morphology, but the molecular profile more similar to an alcinec. So I'll just give a rundown quickly of where we're at with these molecular groups. So my most recent work has been on multiomics analysis, where we integrate whole genome sequencing, RNA sequencing, and DNA methylation array data for a series of 319 pulmonary carcinoids from our lung genomics project. I then apply a theory called Pareto-TI, which is based on the evolutionary theory of task specialization, which is basically a fancy way of generating clusters, which are like molecular groups. So I apply this theory and I identified 4 distinct archetypes, so 4 molecular groups, and these corresponded to the 3 previously reported groups of A1, A2, and B, as well as a 4th archetype, which is colored in pink. And this one contained almost all of our supracarcinoid samples, which are circled in black. And so we're calling this group at the moment the supracarcinoid enriched group. So each of these molecular groups displays differences in clinical and biological features. So I've summarized a few of them here. We have differences in sex, age, location, survival, and there's also particular genetic features and copy number alterations. So the molecular groups are really important because not only do they have some bearing on survival, but they point us to there's differences in the tumor biology. And so this means that we might be able to identify potential targetable biological alterations. So the most important thing about this slide is that you can see in the type section is that in each of the molecular groups, we have a mixture of typical and atypical tumours. So our molecular classification system and the current WHO classification don't really match very well. So in addition to morphology and genomic profiles, other existing techniques such as immunohistochemistry or nuclear medicine may also help us identify and treat distinct groups of lung nets. So the use of immunohistochemistry or IHC to assess the presence of individual prognostic or diagnostic biomarkers in lung nets has been commonly applied in routine pathology practice, but it's restricted to a limited number of targets at the moment, things like Ki67 and somatostatin receptors. So multiple studies have proposed that there might be other prognostic markers for LNETs. So for instance, in this work by Papaxtis et al., they found that the combination of CD44 and OTP staining identifies 3 groups of carcinoids with different prognosis, and that this worked better for prognosticability than separating tumours into typical and atypical. So neuroendocrine neoplasms in other body sites have really benefited from advances in nuclear medicine for diagnosis and treatment, and this is something which hasn't really been explored very much in lung nets. So one example of the utility of this method, One example of the utility of this method is this work by Jindal et al. And they identified significant differences in radiotracer uptake between typical and atypical carcinoids. So we have all these observations now that you can classify samples by their morphology, their molecular group, perhaps immunohistochemistry and nuclear medicine. And it gives us an exciting opportunity to try and develop an optimal approach for diagnosing and treating LNETs. So this brings me to the study aims and hypotheses. So the principal aim of this project is to reconcile the histopathological and the molecular lung net classifications. The hypotheses I will touch on today are as follows. First, that the other proposed systems, classification systems, based on IHC or nuclear medicine, will be consistent with the molecular groups that we've identified of lung nets. And second, that computer vision algorithms will be able to, that we'll use with whole site images, will be able to reconcile these morphological and molecular groups. So this work will be performed mainly on a study group called the Lung Genomics series from our Lung Genomics project. And this is a cohort of lung nets that have been analysed by 6 expert thoracic pathologists. We have 3 sequencing modalities, as well as clinical data and FFPE tissue. Participants from the imaging study will come from the European Neuroendocrine Tumor Society LungNet Task Force. This project is in its very early stages, so I'm going to present a little bit about the methods and some preliminary results that we have so far. So the first step of this project will be to take representative samples from each of these 3 molecular groups that we've identified in this previous multi-omic analysis and perform IHC on a panel of markers. So this will allow us to select markers that best discriminate between these molecular groups. As a first example here, I'm showing the differences in gene expression, so this isn't protein, between molecular groups for a subset of these proposed markers. I know Dr. Dirks is going to present some work after this on the utility of IHC to distinguish prognostic groups of lung nets, so I won't go very much into this, but the results are very promising. And I think it's a good indication that IHC is going to be useful to identify molecular groups when running multi-omics analysis on every single patient is very unrealistic. So following on from this, we will examine patterns of positivity for radiopharmaceuticals through PET scan imaging and assess whether patients cluster based on these patterns of imaging. So we're going to assess whether these clusters correspond to perhaps atypical typical tumours, perhaps they correspond to the molecular groups, or perhaps they identify new clusters with different prognoses. And so this will allow us to identify groups of patients that might respond specifically to different types of therapies. In preparation for this, I've assessed the expression of genes related to selected radiotracers, and we can see already some differences between the molecular groups. So, for example, we have high expression of somatostatin 2 in carcinoids A2 and the cholecystokinin receptor 2 in carcinoids B. So this may mean that these groups are uniquely amenable to radiopharmaceutical delivery through dotatate or CCK2 receptor ligands, respectively. So the second part of this project is to uncover the morphological features of these molecular groups. So we've done this in two ways. Firstly, very simply, I've assessed some enrichment of morphological features that were identified by our 6 expert thoracic pathologists to see if there's any differences between our groups. So at the moment, we have an enrichment of spindle cells in carcinoids A1, conspicuous nucleoli in carcinoids B, and pleural invasion of the supracarcinoid enriched group. So in a much more sophisticated analysis, which has been done by our PhD student, Emily Mathian, she's been training an unsupervised machine learning model based on something called Barlow twin, using H&E stained whole side images. So just a quick explanation of how this works. Basically, the whole side images are cut into teeny tiny pieces called tiles, and it depends on the slide, but roughly 160,000 pieces per image. So these pieces are then run through a model which looks for similarities in morphology in an unsupervised way. So we don't ask it to find cells with a particular morphology, we just say, what can you see? So the model the result of this model is then each sample gets a position along one of 128 vectors. And the principle basically is that samples that are very close along a vector or tiles that are very close together on a vector are more similar in terms of morphology than those further apart. So the vectors are then used to perform something called laden clustering, which is like an easier way to understand basically the output of these results. And it clusters samples into groups called communities based on these vector positions. So these are some of the results. So in plot A, what Emily has done here is plot all the patient tiles in a 2-dimensional representation. Each tile is coloured by the known molecular group of the patient, because these samples come from our multi-omic analysis. In B, the tiles are coloured by their community. And in C, on the x-axis, you can see each community, and then on the y-axis, the proportion of the different molecular groups within these communities. So the communities were found to be able to better predict molecular group than they were to predict tumour type. So for example, we have an area under the curve value of 0.8 for molecular group position and 0.61 for tumour type prediction. So the next step will be to group these, have a look at these communities and have our pathologists try and annotate what is the morphology of these different communities to see if each molecular group does have distinct morphology. So in summary, we have lung nets that can be classified based on the mitotic count and the presence or absence of necrosis into these very important prognostic groups of typical or atypical carcinoid. But they can also be divided into molecular groups which have unique biological features. And importantly, these systems don't really seem to match very well. We have shown evidence that IHC may be able to be used more simply to identify the molecular groups, and that there may be an association between radiotracer uptake and the molecular groups, suggesting some potential targeted therapy options. We also show that unsupervised deep learning algorithms can assist us in identifying the morphological features of these groups. So it's very challenging to try and implement the findings of multi-omic research or molecular analysis into standard clinical practice. So we're hoping that this work will provide the information to overcome these challenges, and that we'll be doing that by including a variety of different methods. So not only the multi-omics analysis, but IHC, nuclear imaging, and deep learning. And our results will hopefully allow for the reconciling of all of these different classification systems to improve the prognosis and management. So lastly, I'd like to thank the leaders of the Rare Cancers Genomics team, Drs. Fall and Fernandez Cuesta, as well as the other members of the team who assisted with this work, particularly Emily Mathian, who has designed and performed all of the deep learning image analysis. I would like to also thank the co-investigators of this project and their teams, Buster Castaño, David Tayeb, Li-Ming Chen, Sylvie Lantejoul, and Thomas Walter. And I'd lastly like to thank the many, many clinical and research centres who provided samples and clinical data for this project. Thank you.
Thank you, Alex, for sharing this very innovative work with us. We have time for maybe one question.
Fantastic talk. I'm going to try to make my question clear. Do you know whether in your transcriptomic subtypes, are your tumor always pure, or can you, if you work those subtypes as components, do you have sometimes tumors with multiple, transcriptomics? Like it's a mixture of different cell types, because that's going to be very important for your AI analysis. I mean, we've seen this in other tumors because they are not pure. When you do the transcriptome on a small area, you get one subtype, but the image you get several, and then that's why your AUC might not be as good as you
I very much understand your question, don't worry. Firstly, these groups are not transcriptomic groups. They're groups that are made from a multi-omics analysis. So it's based on the gene expression profile, the methylation profile, the copy number variant profile, and the mutation and structural variant profiles. And I'm actually in the middle of doing this at the moment. Basically, for about 40 of our samples, we have 2 to 5 pieces of tumor. So what I have to do is take out the original piece from my analysis and put in the new piece and rerun it and see if I have the same group for every single piece of tumor. So I did this in a slightly dodgy way at the moment, which is like a fast way a couple of weeks ago. And it's quite, in the multi-omics analysis, they're quite static. So far I don't really see any samples that actually fit into different groups, but they're certainly in different locations on that molecular map that I showed you. So some patients, all the pieces are like on top of each other in the map, and other times they're a bit further apart. But so far I haven't found any that move. However, in Emily's work, you can see that, it's actually not shown here, but she's been doing DSP analysis. So this is the digital spatial profiling analysis, which is based on protein expression. And she uses this random forest algorithm to try and classify each area of interest from the sample into a molecular group. And most of, for example, the A1 samples, they're always A1, like it's really easy to find them, but the other samples, they move around a little bit. So I do think that, there's going to be some differences. And even in the community analysis, you see some communities are really pure for one type and others are a mix.
Quick question. Since you're relating gene expression to histology, do you think it might be useful to start pushing into the area of three-dimensional transcriptomics to try to understand some sort of spatial and intercellular environment, a component that could fill in some of the missing relationships?
So we have run spatial transcriptomics on 4 of our supercarcinoid samples, but I haven't done I'm not doing the analysis for that, so I don't really know the results. But we are also looking at microenvironment quite heavily, and there's definitely differences between the molecular groups in terms of their microenvironment. They have different proportions of different types of immune cells. So I think this will be an important driver of their differences.
Please come back and tell us about what you find next year.
Thank you.
Thank you.
So it's a great pleasure to introduce the last speaker for this session is Jules Dirks, who's from Maastricht University Medical Center, who will be talking to us about immunohistochemical identification of clinical subtypes and potential therapeutic vulnerabilities in lung carcinoids. Over to you.
Thank you for the kind introduction and giving me the opportunity to speak here. Let me see if I get this working. So my background is I'm a pulmonologist. I'm a resident in pulmonology. I almost finished my training in a few months, and I've been seeing a lot of lung carcinoids patients during my clinics. And I've been working for a while with the group of Sachs and Oates, and we're trying to collaborate on the project to get all these results to clinical practice to get it to my patients. So no disclosures. It's good to talk after her because I think the majority has already been said. When we look at the classification of lung carcinoids or neuroendocrine neoplasms at the moment, we know this separation. So the WHO separates different classes based on the morphology, which utilizes necrosis and the amount of mitosis to separate patients. And we know that this translates to our patient for different prognosis. I would always like to show this. You have a sheep, or it's in fact not a sheep, it's a wolf, simulating a patient with a typical carcinoid. We think it's indolent, but it in fact can metastasize. The atypical carcinoids are much more aggressive, and then we have the neuroendocrine carcinomas, which are really aggressive diseases. Unfortunately, when we look into biopsy specimens of patients, we cannot provide any grading of these tumors. So we can apply it on resection specimens, but in patients who have metastatic disease, we utilize biopsies often, and we can't provide the gradation there. So we looked previously into a group of about 300 patients who had a biopsy specimen and resection specimen. And what you can see here is that from typical to atypical and atypical to typical, it differs on the biopsy specimen and the resection specimen. And that's also why the WHO states that we can't provide this typical atypical separation on a biopsy specimen. However, when I see a patient in clinic, I would like to know what kind of gradation is there, and also because we tend to adjust our treatment in the metastatic setting based on this gradation. So I think we need to improve this for a patient to be able to perform a better gradation on the biopsy specimens, and that's what my project is on. First, we looked into the molecular features of these diseases, and we thought, can we use some of these markers here, as shown by Alexandra, OTP and CD44. When you have a loss of these markers, it's prognostic relevant for patients. And we know that MEN1 is generally associated with a poor prognosis in atypical and typical lung carcinoids. And this likely is mainly informed by the methylation, but also some other processes, while in the neuroendocrine carcinomas, it's generally mutations that cause difference in therapeutic outcome and generally smoking associated. While in our patients, we don't know what is the cause of their disease yet. So this has been shown already also. So I think we're going from a typical atypical classification more to a genomic classification or a transcriptomic classification, which utilizes methylation, expression data, and also some mutations. And then you can see that you in fact have three different groups, so the A1, the A2, and the B, and also potentially some supercarcinoids, but these are harder to identify. And the group from Lyon is not the only group who has shown this. There's also a group from Memorial Sloan Kettering, which has shown exactly the same results, only a few months apart. And when you delve deeper into this data, you see that there are several genes that may be utilized on a biopsy specimen to identify the different molecular subtypes. So here you see data of OTP, ACO1, HNF1 alpha, and also TTF1, S100B, and ANGPTL3. And the same pattern is observed in the group from the Memorial Sloan. So we may need to use these different markers on the biopsy specimens to move this further to our patients. So that was and has been my main aim over the past few years, is to develop an immunohistochemical panel which is applicable on biopsy specimens that we can utilize for a better prognosis and better identification of molecular subgroups on these small tissue samples. So for that, I received an ENETS Translational Research Fellowship 2 years ago to utilize such a panel in a multiplex assay. We're still working on this, and then associate biopsy specimens with resection specimens and see the outcomes there. And from the NETRF, I received a mentored grant last year to look into biopsy specimens in patients with metastatic lung carcinoids and then related to treatment outcomes. So the main treatments that we provide, somatostatin analogs, everolimus, and also chemotherapy. But this is a work in progress, and unfortunately I can't show the data yet here today, but we're still working on it at the moment. So what I will show is how we developed our immunohistochemical panel and why I think this is of relevance to our patients. Here you see the overview, which has been shown previously as well. You can see OTP, ACO1, and HNF1 alpha, and above you see the different groups A1, A2, and the B. And there's a quite sharp distinction between these groups. We then applied this on 15 lung carcinoids on whom we had transcriptomic data, so RNA expression, and we had protein data, and you see that these groups perfectly match. So there's no post-transcriptional processing of at least these three genes to identify the subgroups. We then utilized this panel, so you can see different H-score cutoffs on a large cohort of lung carcinoid patients who had surgically resected, who were treated by surgery. And here are the data. So this immunohistochemistry panel is applied on this group of about 500 patients, and we did a principal component analysis. And again, you see these very sharp clustering of different groups into A1, A2, and B. And on the right side, you see the H-score. So dark red is high expression up to 300, and when it's more the blank color, there is no expression. And you see a sharp difference between the different markers and the different groups. So I think we can utilize this for our practice. We then looked into the prognosis prognosis of these patients. So this is recurrence-free survival. We didn't include death as an outcome, but just recurrence, because a lot of our patients do not die from the disease, or it takes very long. So I think we need to look at recurrence-free survival. Now you see there are some differences for the molecular subgroups, but the WHO grades something different. So it takes out a different biology, and you see the gradation of the WHO is better than the molecular subtypes. However, when you then look into the features of these patients, you see different groups within these classes. So for A1, we generally see it's enriched for elderly patients, elderly female patients. So 80% was female, about 80% was above 50, and they generally were located peripheral. So 50% of all tumors were located peripheral in the lungs. However, when you look at the A2 group, you see a lot of young patients. So 33% of our patients were younger than 40, and these mainly had endobronchial located tumors, so large tumors in the bronchi causing complaints. These patients generally had a more indolent behavior, so 7% did get recurrence of disease, but the majority was local recurrence. And when we then look into the B group, we see this is enriched for males, so 60% is male. It's more aggressive, so 16% was atypical histology. The tumors were larger of size compared to the others, and they had more recurrence. So 1 in 5 had, in fact, a recurrence of disease over time. And what was more striking, when we think of A1, these are patients who have peripheral disease, are elderly, female, and this is something I recognize from my practice from patients who have diffuse neuroendocrine cell hyperplasia. It's enriched for these phenotypes. We then looked for neuroendocrine cell hyperplasia near the tumor, but also in normal lung tissue at distant from the tumor, so a different block. And we found that in fact it's enriched. So A1 has enrichment for this neuroendocrine cell hyperplasia. And also when you look at it in a normal lung tissue, this same enrichment is there, so really separate from the tumor. So I think this is a different kind of disease, or maybe a different cell of origin that we're looking at. And finally, we looked into therapeutic targets. So this is again protein scores for somatostatin receptor type 2A and for DLL3, one upcoming marker, one no marker for treatment. And you see there's a distinct expression. So A1 does not express somatostatin, almost not, but A2 and B have a high expression, while DLL3 is mainly expressed in the A1 subtypes, but not in the A2 and the B. So to me, this tells me that these patients may be treated differently than what we currently have available. So to conclude, my aim is to develop an immunohistochemical panel that would allow prognostic and predictive evaluation of our patients on the biopsy specimen. And thus far, I've shown you that we have established a panel that allows to separate these molecular subtypes, that this panel, separate distinct subgroups and distinct clinical features, which is different from the WHO, and that it expresses different therapeutic targets. So in the future, I think we need to treat our patients differently there. There are still some next steps, so we're comparing our immunohistochemical panel on the biopsy specimens and the resection specimens, and one of the PhDs working on this, Tijmen van Weert, will present these data hopefully during the ENETs. We're going to look into spatial differences, so the question just there, we're comparing primary tumors and metastatic samples to see, do these have a different expression pattern, how is this over time different? We will submit this to the ENETs as well. And finally, we will look into my main question for the NETRF to see metastatic lung carcinoids. Can we apply our pattern on the biopsy specimen there? And do these patients have a different outcome to different treatments? With that, I would like to thank all our co-investigators, my mentors, Annemarie Dingemans and Ernst-Jan Speel, and of course the NETRF for funding our research. Thank you.
Thank you. So if there are any questions, can people make their way to the microphones, and could our other speakers at the same time start making their way up to the platform for the open Q&A?
Thanks, great presentation and excellent interaction between the previous one too. I was curious about some of your markers the OTP, the ASCL1, et cetera. Do you know, is there methylation, or are there any somatic alterations in any of those genes?
Yeah, so one of the PhD students who recently got her PhD looked into this. So for OTP, at least, I know it. For HNF1 alpha, it's still being investigated. But for lung carcinoids, it's mainly methylation-driven. OTP is, in fact, we can't find any mutations, but it's GC enriched, so maybe our methods can't find the mutation in this area of the genome. There are no deletions, rearrangements that we can find for this gene, but we do see a lot of differences in methylation. So there's a paper in Cancers, which has been published in 2022, and you can look up there which different CPG spots are different for OTP, and we think that this is mainly methylation driven. For ACL1, I'm not sure. I think maybe Alexander can answer this data. But this is a very heterogeneous marker, which is difficult to evaluate, so you need to include it with another marker. So ACL1 can be only positive in 2 or 3 or 10 cells, and still you can see this very distinct separation on the immunohistochemical panel. Hope that answers your question.
Brilliant, thank you. If you want to come and join us on the platform, I just wanted to highlight it's no mean feat that in a handful of slides you showed 500 cases with follow-up over 10 years. I mean, that's a phenomenal resource and amount of work, so well done.
Okay, such outstanding talks and such a great way to start this this symposium featuring the technology and sort of the brave new world that's what's coming at us. So we've got about 15 minutes to have open discussion, ask additional questions, but we are trying to make sure that we at least align it with some themes. So I'm going to start with just one theme idea, and that is that with this technology and with everyone's talk, a common feature can be simplified in a simplified manner to say that patients can be kind of divided into categories. In some cases, it's therapy responsive and therapy non-responsive. In some cases, it's angiogenic or not angiogenic. It might be patient-derived or organoid competent xenograph, makeable or not. And then we have in the middle of these multi-omic clouds that you can see these data that are allowing us to segregate cell types. But basically, the question there is what makes a bad case and what makes a good case when you break all that down. So with all this new technology, and each of you did a great job of showing where the advances are headed, I wanted to know what are the key determinants? What are the causable, detectable, attractable mechanisms that are in all this data? And if that's the goal of us to find this, because we're all looking for it, what's holding us back in identifying and leveraging these key factors? Is it the lack of specificity? Is it the noise? What are we missing, and how do we take advantage of this and leverage it to move ourselves forward towards identifying the causes and bringing new therapies to the clinic? And I hope that's something that each of you feel like it recognizes that your own studies are actually addressing in very elegant ways. And I would just open that to discussion for anyone who has a comment.
I do this. I could start just with specifically for lung neuroendocrine tumors. One of the biggest problems is that they're so rare. So it's very difficult to come up with a cohort. I mean, Jules's is amazing. It's 500 samples, but it's not like fresh frozen tissue, right? So you can't just go and do genomic sequencing on 500 samples. So if you're going to do like clinical trials of patients, you need hundreds and hundreds of patients, and you need to know their genomic status. And it's so difficult to build cohorts like this. I mean, we have 319, and these are from like 7 different countries, and has taken years to put together. So I think this is a really big problem in lung nets specifically, because it's so small, the sample size.
Jules, would you like to take a stab at that?
Yeah, of course, I agree on that. And I think what's really nice in the collaboration we had over the past few years is actually we had a different aim for our group, and that at some point we found each other and said, well, we really have this clinical aim and you really have the biological aim. And that really fosters a good collaboration where you can delve deep into the biological background, but then also apply it to clinical practice. And that's really what we're trying to do. I agree it's really rare and we can't do genomics in all cases. I don't think we need to. If you have several really good cohorts which make fair, so you can have the data available to all the researchers, then other people can look into this data and utilize them and then think of some way to utilize them to clinical practice. And I think there, the strong focus should be pointed to that, to get it to clinical practice. And I don't think we need hundreds of patients. If you see a signal, you can make then a good cohort to validate this signal. Oh, that's my opinion.
Emily, I know we have a question from the audience, but one of the things that just really striking about your work is you're really leveraging artificial intelligence in radiology to start to derive determinants of who's going to be responsive to the therapies that you have available now. I know that there's a much broader movement in radiology to just automate, and this is happening across different medical fields, but there's enough data and enough images in existence that basically you can feed it all into a computer and it can look for what's standing out. And these neuroendocrine cancers they're tumors, but they are also neuroendocrinological maladaptations, and they come with a lot of symptoms, and we may not know all the symptoms. Do you imagine that you may be able to expand your studies beyond just kind of seeing where the tumors are with the CT scanning to maybe spotting other unknown or at least partially known peripheral organ or tissue systems that may be showing a phenotype that indicates you've got a problem here? And, I imagine, serotonin overexpression and fibrosis being just one example, but there may be other things going on in the bodies of our patients that this imaging provides the chance to catch, but we've never been able to put that much data analysis into it.
Yeah, I think a really good question. I don't know the answer. One start would be to start looking at whether functional versus non-functional tumors have different features. I think, our field is lagging behind other cancers, but in other cancer types, like in lung cancer, radiomic features have been useful to predict the genomics, which I think is really interesting. Even in neuroendocrine, there are some papers saying that you can non-invasively assess grade. Again, very early days. And one thing I would just say about the limitations, sample size, we need to get these images banked, so the research can be done on larger cohorts. But the other thing is right now, it's a pretty labor-intensive process. For the radiomics itself, you have to segment each tumor that you're going to assess. But this is actually another area that people are looking at ways to develop AI-driven methodology to automate that process. So I think we're getting there, but it's early days in the field right now.
Martin, did you want to come in?
Oh, yes, thank you. So I suppose in terms of thinking of the generalities of it all, one, I think to make a comment on lung nets. Lung nets are 20 to 30% of all neuroendocrine tumors. The problem we have is that they're not coming to the right people. The pathway's wrong. And I think part of what NETRF, part of what NANETs should be doing, what ENETs should be doing, is actually making sure that pathway, like it is for JEPNETs, is heading the right way, because that's 30% of all tumors. So the numbers should be much, much higher. It's our job, actually, of all these organizations to make sure that pathway is improved. And that's getting it to the lung surgeons and the lung community. So that's just one aspect. And sticking on the lung side of things, but maybe pertinent, is there's many clues in here, because you've got atypical carcinoids which are associated with smoking, typical are not. You've got the metastatic disease is so different in typical versus atypical. Atypical may metastasize to the brain, typical tend not to, and you see the prevalence of bone metastasis with lung carcinoids perhaps more with typical than maybe atypical, and liver mets the other way around. And so there's many clues here, hopefully, that can be teased out, which may be applicable across neuroendocrine tumors. And I think the sex aspect, sex hormone aspect of it as well is important, because you've got DIPNEC, 90% women, and then you've got your data as well in terms of coming in terms of the group 1 that you described, although it was a little bit odd that the more benevolent A1 group that was described by Jules didn't have somatostatin type 2 receptor, which you would have expected as well. So it was just some general comments there, but it does have overall themes for the future.
Thank you. And Joe?
Yeah, that would be also a comment. I think especially for lung net, but for other net, I think the time of the frozen biology is over in terms of, I mean, we've done all the omics we could do on frozen sample. That gave us a lot of information. That's a lot of bias because frozen sample is some type of tumor of patient you can operate on. And I think today there's no excuse to move to multi-omic on FFP. I mean, RNA-seq on FFP is beautiful. You can do some deep sequencing on FFP, and I think that will open really the number of samples you can access, and the supracarcinoid, for instance, you would probably find much more of them. And I was wondering, what do you think about the time to move for clinical purpose to FFP-based classification for even some of omic sample?
If you'd like to speak first, and then Jules.
Okay, so FFP is great for example, DNA methylation analysis. I did my PhD in this. And okay, so maybe, yes, we could use some for RNA sequencing. I think that is very much the jury is out on whether you can use FFP tissue for good quality whole genome sequencing. You could probably do it if you're looking for like one specific mutation that you know is like quite common in a particular group of patients, but I'm not sure that I would ever use it for like discovery. But then again, saying that the multi-omics groups that I've shown, they're actually quite reproducible when I just do it with expression methylation data. And so the genetic mutations, because they're so few in lung nets, they're not adding that much more, and what is probably adding a bit more is the copy number profile, and this you don't necessarily need to be doing whole genome sequencing to get a copy number profile, especially in lung nets. The alterations that we're seeing, they're broad, they're arm level gains and losses, so you don't need to be as you don't need to have this fresh frozen tissue. So I think yeah, we could definitely move. Now that we have, I think we've done sequencing on about 100, whole genome sequencing on about 100 lung nets. So maybe we could move to validating our groups in FFP just using expression methylation and maybe some copy number arrays.
I was just going to move on to another theme, so perhaps if you could hold your question to the coffee break. We just have 5 minutes left to bring some of the other presenters in.
Maybe can I make a quick comment about the FFPE? So something we are doing, I'm not sure how many of people in the audience know about this. So the 10x Genomics now has this flex technology, which is not based on 503 UTR, it's probe-based. And we've done it with FFP with 4 samples, and the results we got were really good. So now we're moving into a much bigger cohort. So I think for discovery, we can definitely go back and use FFP and use single cell RNA-seq. It works really well. So it's definitely something I would recommend.
Thank you. I was just going to launch very quickly another theme for the last 5 minutes or so. And I think we've seen this morning 2 fantastic examples where historically we've really struggled to take our translational findings into the clinic, and both Christopher's work and Jules' work, we've seen where we can now potentially use immunohistochemistry to look really quite simply for patients which might have a worse outlook and worse prognosis. So I'm wondering if as a group we should think about trying to do some adjuvant therapy studies again, if we can now potentially use these markers clinically. I was going to open it up to the room and ask Christopher and Jules first along those lines, because I know there have been attempts historically, but I thought I'll just put that back out there. Now that at diagnosis, we can perhaps do a relatively quick test to think about adjuvant therapy, or a trial for adjuvant therapy.
Yeah, that's definitely one of the reasons that we wanted to pursue this and really get a clinical grade assay for ALT. Obviously, I want to talk to others in the room in terms of what kind of trials we might be able to do. But I think one of the major limitations has always been being able to do it in a research setting. I can always do this in my research lab, but to actually be able to translate it. And again, I think we're doing a lot of discovery-based research, which is fantastic. And then at some point, it needs to narrow down to whatever that is. You know, in our case, it was for ALT. But obviously, it would narrow down to other biomarkers that can be done in a clinical setting. And you want to be able to do it in multiple different institutions. So I think that's definitely the next step, and it's something that we're excited about.
Thanks. And Nicholas, do you have any comments around that with the assay that you presented today?
Well, no, I just agree with Dr. Bibb. It's a lot of noise involved, particularly when you're looking at human patients. And then the point is you're trying to sort of stick with it. And, you know, it takes a long time, you know, to look at the specific characteristics of a tumor rather than at that particular time and look at how that patient develops. So that was the whole point of my presentation.
Do all negative tumors, they just not proceed because they are not tumors, or they going in distress? Do they not make telomeres and they just can't go forward?
So actually, we definitely think they are producing telomerase, so their telomeres are maintained through telomerase, so it's another mechanism. Just also to point out that not all negative tumors don't progress. So if you look in primary tumors, about anywhere from 25 to 30% are ALT positive. If you look in metastases, it's anywhere from 70 to 75%. So it's definitely enriched for ALT positivity, but there is another subgroup that are ALT negative that are also progressing. And you can see that in some of the Kaplan-Meier curves. I think those are actually, and we're looking into this, and other groups are too, I think those are just fundamentally different, almost subgroups of tumors, but they are being maintained through telomerase. So I'm not a clinician. So I think that's something that we're starting to look at. Yeah, and I think some of the data that I showed in terms of looking at the small tumors, 2 centimeters or less, I think we need additional biomarkers in that setting in terms of either having surgery or maybe undergoing surveillance at that point. And I think ALT is one of those biomarkers. By no means do I think it's the only biomarker there.
Please, I encourage you all, we encourage you all to engage the speakers in discussion outside of this session. I wanted to just close by mentioning that what I believe that you all are able to appreciate from these outstanding presentations and this outstanding group of individuals here is that we're seeing a transition of high throughput approaches to more creative and innovative applications. What we're learning is starting to take off in more logarithmic fashion. It's increasing in pace. The data we're deriving, it is descriptive and high throughput. But these approaches are hard work. They're expensive. They require infrastructure. They require high levels of computational technology. And they require a lot of personnel. And so I think we'd all agree that this is what NETRF is making possible. And we're grateful to this organization that it's the engine that's driving into this bold new world. So with that, I ask us to thank all of our speakers with a round of applause. We have 15 minutes. Everybody be back at 11:15 sharp.